Hello and welcome to The Explainer. Today, we're going on an incredible journey, deep into the microscopic world that exists inside every single one of us. We're going to take a tour of one of the most amazing factories you could possibly imagine. It's tiny, it's inside every cell in your body, and it is working around the clock to build the very stuff you're made of. We're talking about the incredible process of how proteins get made.
So, have you ever wondered, how does your body take a genetic code, a string of letters, and turn it into something real? How does it build your muscles, your hair, the enzymes that run your body? Well, the answer is a mind-blowing process called translation. Let's dive right in.
Okay, so picture this. A molecular blueprint, which we call messenger RNA or mRNA, arrives on the factory floor. This blueprint is a direct copy of your DNA, and it carries the exact instructions for building one specific protein. And the factory that reads this blueprint, that's the ribosome.
So, what is translation, really? In biological terms, it's the process of linking together these little building blocks called amino acids to form a long chain. We call that chain a polypeptide, which is basically just the technical name for a protein. But here's the absolute key. The order of those amino acids is dictated perfectly by the sequence on the mRNA. It's an incredibly precise conversion of information into a physical thing.
But hold on. Before our factory can even start building, it's got to get its raw materials ready. You can't just have a pile of amino acids lying around and expect a protein to pop into existence. Nope, they need to be prepped for action.
And this prep work happens in two really critical steps. First, the amino acid gets energized in a process called activation. And then it gets loaded onto a special delivery molecule called transfer RNA, or tRNA. We call that step charging. So, step 1, activation. This is all about energy. The cell grabs its universal power source, a molecule you've probably heard of called ATP, and uses it to juice up the amino acid. A special enzyme helps out, and boom, you have an activated amino acid, all fired up and ready for the next stage.
All right, now it's time to load the delivery truck. That energized amino acid gets attached to its very own specific carrier molecule, the tRNA. You can honestly think of tRNA as a tiny, specialized courier that can only carry one specific type of amino acid. Once it's loaded up, we call it a charged tRNA, and it's officially ready to roll out to the ribosome factory.
And now for the main event. With all our building blocks prepped and ready to go, the factory itself starts to assemble right on top of that mRNA blueprint. This is the initiation phase, and this is where the magic really begins.
To get this whole party started, the cell needs to gather all the key players. You've got the two halves that make up the ribosome, the mRNA blueprint, the very first charged tRNA, some special helper proteins called initiation factors, and yep, even more energy in the form of GTP.
Of course, a factory needs to know where to start building. They can't just begin anywhere on the blueprint. In simpler cells, like bacteria, the ribosome looks for a specific tag called the Shine-Dalgarno sequence. But in our more complex cells, it finds a special cap right at the very beginning of the mRNA strand.
The ribosome itself has three important docking stations. There's the A site. A is for arrival. That's where the new amino acids come in. Then there's the P site. P is for polypeptide. That's where the protein chain is built. And finally, the E site for exit, where the used-up couriers leave. Now, to kick things off, the very first tRNA gets to skip the line and binds directly to that middle P site. Okay, now we're in the thick of it, elongation. This is the assembly line in full swing, adding one amino acid after another with absolutely incredible speed and precision. In our cells, this assembly line can add up to 20 amino acids every single second. Just think about that.
So the elongation cycle begins. A new charged tRNA, carrying the very next amino acid dictated by the code, docks right into that now open A site. This is the official arrival dock for all the new building blocks.
And here's where the real magic happens. A peptide bond snaps into place, linking the amino acid at the P site to the new one at the A site. But get this, what's truly amazing is that the catalyst, the thing that makes this reaction happen, isn't a protein. It's actually a piece of the ribosome's own RNA. We call that a ribozyme. How cool is that? So with that new bond formed, the entire ribosome moves one step down the mRNA blueprint. This little shuffle is called translocation. The now empty tRNA in the P site moves to the E site and gets booted out.
Meanwhile, the tRNA holding our growing protein chain slides from the A site over to the P site, and just like that, the A site is open for business again.
And that's the rhythm of the factory. Arrival, bond formation, translocation. It's this beautiful, efficient three-step dance that just repeats over and over and over, building out that protein chain with incredible efficiency.
But this can't go on forever. The assembly line is humming along at full speed, but eventually it's going to hit a stop signal written right into the mRNA blueprint.
These stop signals are just three specific three-letter codes on the mRNA. When the ribosome's A site runs into one of these, UAA, UAG, or UGA, it's like a red light. The factory knows the job is done.
And when this happens, instead of another tRNA carrier showing up, a special protein called a release factor binds to that stop codon. This is the universal signal for pencils down, we're finished.
This release factor acts like a pair of molecular scissors. It cuts the bond holding the finished protein to that very last tRNA. The brand new protein is released, and the entire ribosome factory disassembles, all ready to find a new blueprint and start a new project.
Oh, and one last little detail for you. The mRNA blueprint isn't all code for protein. It has these buffer zones at the very beginning and the very end called untranslated regions, or UTRs. They don't actually get turned into the protein itself, but they are absolutely essential for helping the ribosome get started and work efficiently.
So to wrap it all up, just remember this. Every single protein in your body is built by precisely translating a genetic message. Information becomes function, code becomes life. And this incredible accuracy is so important. Just one mistake in this process can lead to disease. It really makes you appreciate that this happens correctly trillions of times a second inside of you.
This microscopic assembly line is always at work, building everything that makes you, well, you. It's repairing, it's rebuilding, it's responding to the world, one single protein at a time. It is the fundamental process that takes the code of life and turns it into life itself.